Welcome to Science with Dr. T. This week, we'll be learning about amino acid characteristics. Amino acids are the building blocks of proteins. They each contain an amino group, a carboxyl group, and a side chain connected to a central carbon atom. The side chain, or R group, is what makes each amino acid unique. To form a protein, the carboxyl group of amino acid 1 and the amine group on amino acid 2 undergo a condensation reaction. It's a condensation reaction because you lose a water molecule. After the reaction, a peptide bond is formed between the carboxyl carbon and the nitrogen of the amine group. Once the polypeptide chain forms, the amino group is considered the beginning, and the carboxyl group is the end of the protein, similar to the 5' and 3' ends of nucleic acids. There are 20 common amino acids that are grouped by the characteristics of their side chains, which are highlighted in light blue here. Nonpolar aliphatic amino acids have straight side chains composed mainly of carbon and hydrogen, making them hydrophobic or water-fearing, and tending to cluster together in the interior of proteins. Polar uncharged amino acids have side chains with polar functional groups like hydroxyl groups, OH in serine and threonine, or amide groups, NH2 in asparagine and glutamine, that can participate in hydrogen bonding with water molecules, making them polar, but they do not carry a net charge, hence uncharged. Because they are water-loving, they tend to be found on the surface of proteins. Positively charged amino acids are considered basic amino acids due to their positive charge at physiological pH levels. These amino acids have side chains containing nitrogen atoms, which readily accept protons, giving them a positive charge. Negatively charged amino acids are considered acidic amino acids and carry a negative charge at a neutral pH of 7. They have carboxyl groups in their side chains, which readily lose a proton at physiological pH, resulting in a negative charge. Both negative and positively charged amino acids are hydrophilic, so tend to be on the surface of proteins. Nonpolar aromatic amino acids have side chains made up of carbon and hydrogen, like the nonpolar aliphatic amino acids. But these are aromatic because they contain a 6-carbon benzene ring. These groups have very different characteristics. So you can imagine that when a missense mutation happens, that's when one amino acid is changed for a different amino acid, it could lead to damaging effects. You should definitely be familiar with the structure of each amino acid, as well as the 3-letter and 1-letter abbreviations. Now, let's put these concepts into practice. If this is the last amino acid in a polypeptide chain, which part or parts will be involved in a peptide bond? So what do we know? We know the amino group and the carboxyl group of an amino acid forms peptide bonds. So that means we can cross out C and D because these are the side chain and the central carbon. So we know we don't need those. And the question says this is the last amino acid in the chain. So that means the carboxyl group will be free because it's the end. So we can cross out B because we're looking for what's going to be involved in a peptide bond. That leaves us with A, the amine group, which will be involved in a peptide bond with the next to last amino acid. Let's try another problem. A DNA mutation causes a glutamate to be exchanged for a valine in a protein. Would this affect the protein? Let's compare each amino acid. Glutamate is a negatively charged amino acid, which means it's hydrophilic. water loving, and it tends to be on the surface of a protein. Valine side chain is made up of carbons and hydrogens. So it is nonpolar, which means it is hydrophobic and tends to cluster on the interior of a protein. So with this mutation, you have an amino acid on the surface, that valine, that does not like water and wants to hide in the interior of a protein. These are very different characteristics that lead to changing the makeup of the protein and the interactions the protein can make. So, yes, this would affect the protein. And this exact mutation is what causes sickle cell disease. The mutation happens in the gene for a protein in red blood cells called hemoglobin. So you have normal hemoglobin, then in sickle cells, the hemoglobin clumps together to try to hide that valine that has been exposed on the surface of the protein. These hemoglobin clumps create long strands of rigid hemoglobin that cause the sickle shape of the red blood cell. I hope this has been helpful. Remember to practice on your own with a few different scenarios until you get the hang of it. Thank you for tuning in. Remember to like our videos, subscribe to the channel, and share. Next time, we'll look at protein structure.